Assalamu alaikum. I'm Tanya Noreen, and on behalf of Shifa Center of Professional Excellence, I welcome you all on this seminar today. Without further delay, I am going to hand over the webinar to Dr. Ali Talha Khalil. So, Dr. Ali, I guess you are with us. Can you hear us?
Thank you, Tanya. I'm here definitely. Thank you, everyone, and good morning, everyone. So, are we supposed to get started, Tanya?
Yes, sir. Yes, sir. Over to you.
Thank you. So, again, good morning, everyone. So today, just a brief about myself. I am Dr. Talha Khalil, and I am working as an assistant professor of molecular biology and one of the leading hospitals in Peshawar, even in Asia as well. And I'm also fellow of the National Chair in Nanosciences and Nanotechnology, fellow of the Nano-Acclimate and member of the Asian Council of Science and Data. So the topic that we will be discussing today more or less relates to ethics and the dual use of the technology. And then the topic that I've chosen is ethics and dual use research in terms of modern biology, and we will be discussing some case studies and what are our responsibilities as a scientist and as a researcher. I'm trying to. Okay, so just before starting initially, let's understand what is ethics and what is morality. They are the two terms that sometimes people use as interchangeably, but they are different. What ethics questions the morality of this. We are researchers, and we are concerned with the research ethics related to that. What is morality? Morality are the principles concerning the distinction between morality tells you what is right and what is wrong, what represents a good behavior and what represents a bad behavior, what characterizes something is good and what characterizes a bad behavior or a bad thing. Well, ethics defines the morally correct and morally incorrect behavior. So, ethics are the moral principles. So, that by definition, moral principles that govern a certain behavior or the conducting of an activity. So it is the morality that tells you what is right and what is wrong, and when there is a set of the principles that you follow, that usually is termed as ethics. Research ethics are the moral principles that guide researchers, and how do they guide researchers? There are the guidelines, and how do they guide us? Conduct and report research without deception, without any falsification, without any fabrication, for example, avoiding plagiarism, intention. Then they have some certain defined principles. For example, there is the maleficence, there is the non-maleficence, there is the beneficence. So what it tells us that avoid something that's not good for research, for example, deception, for example. And then intention to harm the participants of the study or members of the society, knowingly or unknowingly. So what research should be that it should not harm, it should not become a source of harm to the society. How scientific research is performed at research institutions. Now, how do you perform the research? How do you follow the guidelines, and how do you follow the the correctness of the research? And how scientific research is disseminated, for example, when it comes to publications, and how do you avoid academic misconduct and practicing of the ethical guidelines? So, 21st century is a century of biology. We are a biologist, and I am addressing to more the people who are actually involved in the health sciences directly or indirectly. So there was in the 19th century, we had the age of engineering, and there was some engineering field accomplished in the 19th century, and that was followed by the chemistry and physics in the 20th century. And now we do have the 21st century that we call it the age of biology. And we will see how it is actually the age of biology. And we will be discussing some of the application of the current biological research, especially in health science. What do we need now in this age of the biology? That we need some smart scientists smart people who develop technology, and there is a need of the smart politicians who do the policy making according to that. And of course, we need smart citizens that adopt that adopt to the new technology and that adopt to the new policy. So why I was talking about the applications of the biology, and there is one example for there is the wonderful concept of biomimicry, in which you mimic the or copy some natural process or some biological process or even biological organisms or some structure. For there is one example that chocolate antibacterial technology takes inspiration. It's based on the material that the skin of the shark. For example, the shark skin, due to its topology, due to its topography, bears some antibacterial characteristics. So now, what material scientists have done actually, that they have created similar materials for healthcare research, so similar materials that can be applied that have the similar structure that is of the shark skin, and they have the antibacterial properties. So in a way, they mimic the microstructures of the shark skin to avoid and for conferring the antibacterial properties to the skin to the any materials that they are developed. And another example for these are bandages created using the biomimicry or using this wonderful concept that there was a team of MIT and Mayo Clinic. They created a medical paste that intend to heal the internal bleeding, and that was much faster than the natural biology of human beings. And then recently, there is one more development that there is the graphene-based photonic crystals. Graphene is considered as one of the wonder material of the 21st century. So they have created some photonic crystals. Photonic basically what it means that they give some different colors. So when they functionalize it, what it is expected that once it is functionalized with some biomolecules or some enzymes, it can give you a very fast detection. It will be just like coloring, changing color of a piece of feather, for example. In a sense, we will be able to diagnose the disease very effectively and very rapidly. And then you have the stem cell therapy. Stem cell is one of the in fact living area of research, and it is actually redefining the healthcare at all levels. So there is one disease that is we call it the age-related macular degeneration, and it's related to the eye loss, the vision loss. There are some RPE cells, the retinal pigmented cells that they die, and eventually what happens that you begin to lose loss of vision. And what's one of the leading cause of the vision loss, especially in elderly people. So with the help of stem cell technology, now we can create. You can pick the blood from the person, from the patient, and grow it in the lab and differentiate it in the sense that it becomes through stem cell technology, it becomes the RPE cells and patch it and decorate them in a sense on the patch and then surgically implant them. And what happens what happened actually that the people with who were lost their vision now have their vision. But in the context and on the other side of the application, what is actually our main focus is the dual side of the biology, or the dual side of the health advances in health sciences and biology raises a dual use dilemma. What is that actually dual use dilemma? The dual use dilemma arises in the context of research in the biological and other sciences when there is actually a particular technology, a particular process, or a particular thing that can be used for beneficial purposes, but the same can be applied, or the same can be in fact misapplied for some nefarious or vicious or criminal purposes. So that's the dual use. That's the dual use dilemma, and that's why they have coined the term the dual use, and we usually refer it as a dual use research concept. I'll be talking, for example, there is the technologies and factors now available that relates to the that constitutes the set of the techniques that allow you extend the age of the people. And there is a question that is for you, and I hope I'll get some answers at the end of this webinar. That should we allow the life extending technology? Should we allow it? Should we allow it? And we will be discussing it further. There is one scientist who actually estimated that if you the aging relates to the diseases, and if you prevent the 50% of the diseases, then the life expectancy can become as high as 150 years. And if you prevent the medically preventable conditions, 90%, for example, then you can live approximately for 500 or something like 500 years. And at 99%, when you solve every medical problem almost, then you can live up to 1,000 years. And we have the current science. That's why we say that this is the age of the biology, because now we have techniques like genetic engineering, like, for example, stem biology. We will be talking about that. That you can actually prevent all the medically preventable disease causes. How can you do that? Cure diseases, stop the process, that can be one strategy. And the other is, for example, you can coat the organs or something with some superior material so that do not get degraded or something. And then you can keep on having technology available. For example, tissue engineering, stem biology, that you can kept on replacing your body organs. And there is the strategies for engineered negligible senescence, which actually slows down the process of the aging, slows down the process of and enhance the repair of the damaged tissue. And there is one more another concept related to mind uploading. What is actually mind uploading is to scan, it involves the scanning of your brain and copy it to some computer or computational substrates. And what scientists believe that by 2045, humans will be able to upload their brains to the digital world. And there is another concept related to cryonics. At cryogenic temperatures, there will be no alteration in the biological tissues. Those who know what is cryobiology or cryogenics, it is some kind of freeze treatment of the organs or the body that allows you to preserve. A complete body organ can be preserved for hundreds and thousands of years, and no longer future medicines will be available. I'll be talking about that in the slide. What is the ethical issue in that? Like for example, we are discussing the ethics. We are discussing the dual use of the technology. At the first phase, we discussed how can or how far the biology can reach in terms of application. But on the other hand, there is a dual side to it. What happens, for example, in cryobiology or cryonics, if you are able to preserve someone or the whole body, so it leaves the loved one in oblivion. For example, there is nothing, but he is just he is just some kind of a sleep or some kind of oblivion kind of thing. And what will it be like to acclimatize in the society? For example, when you revert or when you able to kind of resurrect them back to the life. So, for example, if you allow this procedure after 100 years, if somebody is made to observe for 100 years, and after that, it is he or she is resurrected, so he will be in the stranger land because nobody, or he will know nothing. For example, where are his relatives or her relatives, or where is and what about the massive knowledge disconnect? Of course, science and technology moves at a rapid pace, and if you are able to resurrect him after 100 years, so God knows what will be the technology at that time. And what if your family disowned you, or if they migrated to some other place, another issue, an ethical dilemma? How does one just plug in back? It is just like it can't be like a USB kind of a thing that you just plug and play. What if what now? If some organization or some company has allowed the procedure to cryopreserve the whole body, And what after 100 years, for example, if the company does not exist, then what will happen? It can become, for example, bankrupted. And what if the procedure is only partly successful and you do not come back as a useful member of the society for another issue, and who will be responsible for that? So just a practical example of it. Of cryobiology. A 14-year-old dying girl. She was a cancer patient, and she filed a case in UK High Court to allow her to be cryopreserved, and eventually she won the case. So now she is in fact cryopreserved in one of the facilities in the United States, and along her there are 4 or 5 other people, and in the hope, they are cryopreserved in the hope that someday, for example, she faces cancer, so after 100 years or 150 years, they will have the treatment available, and then they can, come back to the world in a sense, and cure the disease. So, and on the lab level and on the minor level, the cryopreservation has been done a lot in terms of on kidneys and brain, and we are later transplanted successfully into the animal. And then you can go for the body partnership, like I have told you that sample biology through material sciences or biomaterials, you can be able to create virtually every part of your body. So that then if some part of the body becomes aged or becomes kind of a defective, then you can replace it with the artificial one. So what are the advantages? For example, if individual brain will be transplanted from her aging body, then needs the body grown from her own tissue. So that leads to the stem cell biology. And artificial organs will be implanted using nano devices, and there will be, for example, further improved version of the body, in a sense that it will be, added with nanotechnological tools that will allow less glucose consumption and less energy consumption in a sense. What are the on the downside? What are the issues related to that? It can be the technology is evolving and evolving. Now we have bionics and some after down the road, some years later, you will be able to have some superhero kind of characteristics or something, being superior or become a superhero. And what is the problem is that organ forming and stockpiling. What if somebody use the technology for creating organs only, and then discarding them, discarding those which are not true. And we draw the line between something apart versus a clone body, and are we doing or are interval going towards spare part human being? Mind approach. Uploading a human brain means scanning all of its fields and then copying them into a suitably powerful computation substrate. In effect, it affects a new and equally valid version. A digital you is created in the computer world. Your brain is scanned through every bits and pieces of your brain is scanned, and that is duplicated in the computational world. The person's mind and memories, emotions, and personality would be duplicated. What are the issues? At which point of time do one perform the procedure? Should we be allowing only at that, only when somebody is going to die and he uploads all his brain to the computer? There are many kind of a science fiction movies related to that as well. And how does society and family prepare for a digital version of it? What if there are like a software, there are updates, version 1, version 2, and so on? How are we going to manage that? And what if someone accidentally or intentionally deletes the upload? Then that would term as killing. Again, a problem that you have your all data copied in some computer, your all brain scans and digital version of you, and somebody intentionally or non-intentionally deletes it. So what will be the definition of in fact killing in that sense? And what is the definition of single? for example, how do you define being old? Where is the definition line? And how it should change if someone know that he or she is living for extended duration. So somebody is not going to die, and he becomes, some kind of a crazy kind of a person. And funding priorities is another issue. Are we supposed to go into fund it? And there is a societal divide because all of these expect that all of technology would be in fact very costly and will be afforded by only the rich people. So it's still in a sense data divided in a society. Eugenics, another concept related to the genetic quality. So what is eugenics? The selection of desired heritable characteristics in order to improve future generations, typically in reference to the human being. So it is in fact the selection of the good characters, good genetic characters, so as to improve the future generation. What are the conception that there is one form of the eugenics known as the positive eugenics, and the other form is known as the negative eugenics. When you encourage the good genes, when you have some desirable traits, for example, it can be related to some tallness or some colors or something. So there is the desirable trait maybe for someone, and then you allow them to flow in the all of the society. And then you have the negative eugenics that refers to efforts aimed at decreasing the undesirable traits. There are maybe some undesirable traits, and you discourage the bad genes in negative eugenics. So how do you do that? How is it possible? Are you post sterilization or abortion, or you are putting some marriage restrictions, for example, to discourage some bad genes and to allow good genes within the society or within the community? Eugenics is a social responsibility and a selfless stand, of society meant to create healthier and more intelligent people, resources and less human suffering. What are maybe on the negative side? Eugenics that is based on pseudoscience is to discriminate. Eugenics is not properly used or not properly applied. It can cause some serious difficulties. And do we have a middle ground? This is a question for you, or all the you can answer that. What are some other ethical concerns related to that? So, what if there is some sort of coercion or some kind of force applied, that interfere with the right to reproduce without interference? That you allow some selective kind of marriages or some selective kind of things to allow the good genes and the bad genes. And what characterize perfection in that sense? Who defines the perfection? What is perfection? That in turn, that attribute is also questioned. That what is actually for some people the perfection would be, for example, in the sense of doing eyes or two eyes, somebody will say no, it's not like that. So how are you supposed to manage that? And equality, fundamental social inequality, and you will be creating a genetic overclass, which ultimately will create problems. And equity and fairness, how to ensure equal opportunities for those. So whatever the technology is available, it should be equally distributed and equally and fairly distributed. What is there is another issue, for example, that's related to the consent process. For example, there is a baby, he or she cannot detect any aspect of his or her upbringing, but he or she can never decide on the genes that are chosen for it. For example, if you have chosen the eugenics and the eugenics, you have chosen something like blue eyes, for example, and but in turns, what the baby is born, and he tells you, no, this is not. I should have black eyes or something like that. And just controlled by one human over another is incompatible. And this is really, and it actually questions the inherent nature, the unpredictable nature of our genome that we are having. So this is another problem. And And there are other issues with the modern science, human cloning, synthetic biology, genetic engineering, and nanobiotechnology. The combination of all these convergent technologies can lead to get many benefits, but at the same time, on the dual side, we need to regulate them in a sense that the benefits are maximized and the risks are minimized. And there are some other studies in the recent times. There was this Spanish flu, if you remember. We are now passing through the pandemic, but in 1918 we often heard a lot the Spanish flu, and it was the deadliest at that time. Now what happens is in one of the works actually they have resynthesized the Spanish flu virus, and they have they have now the full sequence of the Spanish flu virus has been published, and further research on the sequence sequences of the same. It is actually what happened that they have created the same 8 gene element of the Spanish influenza virus that was was present in 1915. And interestingly, the virus which was created as a result of this research was found to be more harmful to chickens than any other flu virus. When tested in mice, it was some kind of very lethal that it killed more quickly than any other flu virus ever tested. So on the one side, the world is actually scientists are trying to eradicate such kind of viruses, but on the other side, they are actually some scientists are actually creating it. So that is dilemma. So a brilliant piece of science is at the same time a recipe for a disaster. Like for example, like if the Spanish flu sequence is available, freely available, what happens if some rogue scientists or some wicked or some criminal people can, with the help of some few little bit of funding, in a sense, they can create the virus. And they can use them as biological weapons. The Spanish flu case underlies the need for biosecurity regulation to prevent the proliferation of particularly dangerous knowledge. We will be talking on that in this later section. And then it is not, and publishing of such information in an open journal is really making a way for other people to read it, and they can. We call these days the biotechnology, garage biotechnology. With the help of the tools and the help of some limited kind of setup, we can actually create the virus through synthetic biology or through such. And there was another case that relates to the polio virus, that has sparked this debate because the scientists have created this polio. Scientists have created, not recreated, in fact, they have created the polio virus from the genome, the map, the DNA sequences available on the internet. And then it was simple, just they have purchased some. You can purchase the DNA. You can purchase order the sequence of the DNA, and you stitch that, and you have created a whole new virus. And what resulted in that polio virus was equally dangerous as it paralyzed and killed the mice. So what is the issue in that? If you are creating, for example, in Pakistan, Afghanistan, we are trying to eradicate the polio virus. At the one hand, people are trying to eradicate it. And on the other hand, people are trying to recreate it. So this is a dilemma that needs to be discussed on now. And another case relates to vaccine resistant mousepox virus. That mousepox virus. That they have been in one of the research, they are trying to insert the interleukin gene. They were studying the infertility, and what they found out that they have created some kind of mousepox virus that actually was resistant to the that actually was more lethal as compared to earlier. And rewriting the genetic pool, scientists experiment that relates to the genetic engineering scientists experiments to alter the genes of human embryos so that resulting babies are immune to the immunodeficiency virus. That's related to the CRISPR-Cas technology, that very precise kind of genetic engineering technology. And it has some safety issues, and we'll be here how do they have done that? That there is some a receptor that allows the entry of the virus to the cell. And what scientists have done, that they have deactivated that receptor, and eventually what happened is that they have created the HIV resistant babies in a sense. But that same CCR5 receptor is also in a sense is very important in the sense that it allows you to fight against some other infection like the West Nile virus. So in a sense, it has created this dilemma that on the one side you have created an immune babies to HIV, but on the other side they are becoming more vulnerable to the West Nile virus and other infections. So this is a problem, and then you do not have this long-term unintended consequences of genetic insurance. So you actually do not have any idea what in the future it will have, what will be the consequences. And then you have the designer babies, like the genetic engineering through techniques. We can now have design the cosmetic drugs. So there are issues of affordability. And again, this will create another divide of treatment. And then there is the issue of the open science. What is an open science? The open science umbrella term that to characterize scientific research in terms of reproducibility and reliability, usability, replicability, and transparency and rigor. So, an open science is a flavor of openness, that whatever research you are doing, it should be open to all. Whatever the techniques you have used, whatever the analysis methods you have used, whatever the participants that you have included in your study, and it is getting popularity in the scientific community. But on the other hand, it has some issues as well. Like this, the open access models are costly, and then you have actually anonymity and confidentiality issues of the participants. For example, whenever you are doing some study, you get the consent from the participants. So in a sense, if you open it to everyone, so you actually are questioning the confidentiality and anonymity principles of it. And then like we have studied some recent examples like the synthesis of the polio virus or the synthesis of the smallpox virus. So if this information is available on the internet freely, so that will have consequences. That will have and it can maybe used by some people for the cricket or for military. Lastly, about suggestions that what can we do? What can be the level of the governance regarding the tool use of this, the tool use of this? So it is ultimately up to the individual scientist that what he or she publishes, what is sensitive, he or she he should be able to decide that what is the sensitive data that can be misused by the people and should not be available in public domain. And the research institutions regulation within the institute, they should regulate and review for any dual use of the technology or the research. And the professional society, the academic publishers, for example, the editors and the reviewers also be capable enough to censor some information that is sensitive, that may be used for some nefarious or religious purposes. And then the national governments and funders and international governments that gives you some awareness related to that. So lastly, about the conclusion, like about the dual use, like it's just like a knife, and a knife can be used to for useful purposes to cut an apple or something, or and can be used for some wicked purposes. So progress in science should not be stopped If you are not going to do it, somebody else will do it. Better you do it, but regulate it in a proper way. And responsibility and awareness, science communication and oversight is important, and we should be taking care related. We should be taking care related to the science communication and publication, and identify what are the sensitivities of that, and we should conduct on and off oversight of the issue. So thank you very much. I am done here. Over to the host of the host of the webinar.
Thank you, Dr. Talha. We have some questions with this. So the first question is, being a health professional, how would you take it as it is complying with bioethics? Getting shots of COVID-19 vaccination is made mandatory for the whole citizens of a country in Pakistan without taking their consent, as Consino was still not approved by FDA.
I tried to read this question. Being a health professional, how would you take it? Sorry, I'm not able to, see the question or something. Like what we can do is, we can regulate ourselves. That's the like my answer would be that like how being a healthcare professional or being a health professional, how would you take it as it's complying with the bioethics? So what we can do maybe regulate ourselves. We can regulate ourselves. We should know our responsibilities. So we should know what is the responsible conduct of scientists. We should know what is sensitive and what should be published and what should not be published, for example. So this is the things that that self-regulation can help us in that. And about the second part related to the COVID vaccination, there is you have to everybody has the right, and there is one issue that rights of not being infected. For example, if you are not vaccinating yourself or somebody is not vaccinating his or her, so he becomes eventually a larger threat to the he becomes a larger threat to the society itself. So in a sense, maybe that's a problem. So vaccination in that is mandatory, and it should be done. Regarding the question number 2,
okay, we have another question. Can you give examples of dual use of research case studies for us to read?
Yes, sure. I can share with you even this presentation, and I can send you some. I can send you some links, some related links, some very good case studies that you will in a better position to understand it, and I'll share some research.
So the learners which we have with us right now, do you have any other questions? You can write in Q&A box. Okay, we have another question. As bioethics is an emerging discipline, what steps would you suggest government to make it complementary in medical education as a core subject in Pakistan, though it is already practiced in foreign countries?
I think very good question. That of course we should build from. We should start it, by teaching it to our undergraduate students, and there are they are in fact getting it. for example, in life sciences, there is a mandatory course for bioethics in the HSC approved curriculum. So I think things are moving in a good direction in that sense. And it should have to be, an important aspect for the medical students too, in the MBBS curriculum as well.
Okay, I guess these are the questions. So thank you, Dr. Talha. Thank you very much for amazing session. Hope our learners learned a lot. So as we always say that this is not the end, this is the start of new journey. We will be sending the feedback forms on your emails, and write us back and let us know about the session, and on your recommendations, we will be asking Dr. Talha to come again and share his expertise, his knowledge with us again on the topics which will be recommended by all of you. So thank you very much. Thank you, Dr. Talha. Thank you once again. Okay. Thank you, Ji. Khuda Hafiz.